Hello and welcome to this bite-sized bio web seminar, which is sponsored by Biosearch Technologies. I'm Nick Oswald and I'll be chairing today's session, which is titled Detection of Long Non-Coding RNAs by Stellaris FISH Probes. It's being presented today by Dr. Arturo Orjalo, who is a scientist at Biosearch Technologies. Arturo has a broad molecular and cell biology background. He has a PhD from UC San Diego and his postdoc work took place at the Buck Institute for Research on Aging. Arturo is now focused on the development of Stellaris technology for Biosearch Technologies. And so now over to Arturo for the presentation.
Thank you very much, Nick. Hello, everyone. As you can see from the title, today I'm going to talk about a simple yet powerful tool from Biosearch Technologies called Stellaris FISH probes that allows for the detection and visualization of long non-coding RNAs at the single cell level. So just to give you an idea of what I'll be talking about today, just the agenda, I'll first start off with the background of long non-coding RNA mechanisms of gene regulation. Then I'll follow that up with the methodology and the application of Stellaris FISH. And then I'll proceed to, well, I should also emphasize that because Stellaris FISH was initially designed to visualize messenger RNAs at the single cell level, a lot of this initial studies and data were done using messenger RNAs. But I should just remind you that whatever you see today is directly applicable to the to studying long non-coding RNAs. And so, and then in the end, I'll talk about the examples, show you some examples of long non-coding RNAs as visualized by Stellaris FISH. So, long non-coding RNAs. So the eukaryotic genome transcribes up to 90% of its genomic DNA, but interestingly, only 1 to 2% of these transcripts actually encode for protein. So just to put this in perspective, we have about the same number of protein coding genes as the laboratory worm. We have 23,000 and the worm has 20,000. Now, the vast majority of these transcripts are non-coding RNAs. And the largest subset of these non-coding RNAs are long non-coding RNAs, which have been arbitrarily set at greater than 200 nucleotides in length. Now, functionally, these long non-coding RNAs have implicated in embryonic stem cell pluripotency, cell cycle regulation, and as well as diseases such as cancer. But overall, long non-coding RNA function and activity still remains poorly understood. Just to show you demonstrate or explain some of the dynamics of long non-coding RNAs, I want to show you how long non-coding RNAs regulate gene expression, which falls into three broad categories through epigenetic silencing, transcriptional regulation, as well as through post-transcriptional regulation. So in this one example, or I should say in the examples for epigenetic silencing, on the left, we have one example of a long non-coding RNA, HOTAIR. It stands for the Hox transcript antisense RNA. It actually is the first long non-coding RNA expressed on one chromosome, but mediating its effects on another chromosome. In this case, HOTAIR coordinates the recruitment of the polycomb repressive complex 2, shown here on the left, as well as the lysine-specific demethylase 1 complex. And what it does, it acts as a scaffold and coordinates the demethylation of histone 3 lysine 27 methylation, as well as demethylation of histone 3 lysine 4. Which leads to the repression of the HOXD locus on chromosome 2, HOTAIR, which is expressed on chromosome 12 in the HOXD locus. Now, on the right, long non-coding RNAs can also repress transcription in CIS. And we have here XIST and ANRIL. Now, although they are here on the same on the same cartoon, they do function on different chromosomes but are mechanistically similar. Now, XIST, it is actually one of the most dramatic examples of long non-coding RNA-mediated chromatin regulation. It actually facilitates the inactivation of almost the entire X chromosomes, at least one of them in mammalian females. Now, this is obviously important for dosage equivalence between males and females. So what XIST does, it will, it recruits the PRC1 and PRC2 on the X chromosome. Which then facilitates the ubiquitination of histone 2A, lysine 119, as well as the methylation of histone 3, lysine 27. Now, similarly, we have ANRIL, which is expressed on the chromosome 9. It also recruits PRC2 and PRC1, which leads to methylation of histone 3, lysine 27, and silencing of the IncRNA RF locus on chromosome 9. Another means by which long non-coding RNAs regulate gene expression is through transcriptional regulation. In this example, we have EVF2. Its official gene name is DLX6AS1. It actually is expressed in this conserved region labeled I1 and 2 on the DLX5 and DLX locus. And in conjunction with the homeobox protein DLX2, it actually upregulates the NX to activate transcription from the DLX5 and DLX6 locus. In the final example, long non-coding RNAs have been shown to regulate gene expression at the post-transcriptional level. In this example, we have NRON, which stands for non-protein coding repressor of nuclear activity T cells. So NRON actually controls the subcellular localization of certain transcription factor, NFAT. It will basically inhibit and interact with the members of the important beta superfamily, which is important for the import of NFAT, activated NFAT, into the nucleus, so that it can actually upregulate genes that it's specific to. So the question now is, what is Stellaris FISH? So Stellaris FISH is an RNA detection method that enables the detection, localization, and quantification of individual RNA molecules at the cellular level. Now, again, I do want to remind you that the data that you're going to see shortly was done using messenger RNA, because this was originally designed to look at messenger RNA. But Stellaris FISH is translated very well at looking at long non-coding RNAs, and you should just keep that in your mind, keep that in your head, that whatever you see here was directly applicable to long non-coding RNAs. Now, as far as the Stellaris FISH protocol, it's a very, very simple protocol. It's quite comparable to immunofluorescence. Now, in this protocol for adherent cells, the cells undergo a 10-minute fixation process in 4% formalin. Then the cells are permeabilized for at least an hour in 70% ethanol. And then the cells are then hybridized with the Stellaris FISH probes in as little as 4 hours. And how does this work is that these FISH probes are 20-mers, and they are single-labeled with your fluorophore of choice. And the idea is that these single-labeled 20-mers will tile along your messenger RNA, and the total fluorescence from these individual 20-mers will create a single diffraction-limited spot corresponding to one RNA molecule, one individual RNA molecule. Now, after the excess FISH probes are washed away, these can be processed and imaged on a normal standard fluorescent microscope, whether it's upright or inverted, non-confocal. And just example on the far right, you can just actually see spots which, again, it corresponds to a single molecule of RNA. Now, what we have on actually the Biosearch website is a very user-friendly Stellaris probe designer. And in this example, I am creating a probe set to the long non-coding RNA NEAT1. and the site will actually prompt you for the probe set name. And in this case, I named it NEAT1 Q570, which is one of the Quasar 570, which is actually one of the Biosearch proprietary dyes, which is also a Psi3 analog. And I labeled it AO, which are my initials. And then in the second line, we have gene name. I just, for convenience, I named it NEAT1. And under organism, I have it human. Now, if as you can see on the left, if the menu was opened up in its entirety, you have the choices of either human, mouse, rat, Drosophila, C. elegans, and other. Now, these are the major system models that are being used today. But if you were to use when it comes to, well, I should say when it comes to choosing the masking level, except for other, you'll start off at the level 5, which is the most stringent, and then you would proceed now to move down until you have at least, I would say at least 25 FISH probes for your target RNA. Now, the number of probes, has a default of 48, default of 20 nucleotides long, and a spacing of 2 nucleotides. And on the right is where you paste your RNA sequence, whether it's long non-coding RNA or a messenger RNA. So after you click on Find Probes, as the output, then you'll actually see the individual Stellaris FISH probes. In this case, I'm only showing 27 of the 48 FISH probes that were generated to NEAT1. And then, which is convenient on the right, you have the area to which these FISH probes bind. So it's just a nice way to see where exactly, these computer-generated FISH probes design your, bound to your RNA of interest. Now, just to give you an example of what Stellaris FISH can do, I will show you this is an image of the long non-coding RNAs, NEAT1 and MALAT1. You can see the very robust signal. These RNAs are actually very much highly expressed in mammalian cells. So I do want to point out though that we can detect and visualize long non-coding RNAs that are much less highly expressed. But before I do that, I do want to discuss the methodology and the applications of Stellaris FISH in the context of messenger RNA, again, because these studies were initially done in messenger RNA. So as a proof of principle, the Stellaris FISH probes were initially designed to the messenger RNA of GAPDH. On the graphic on the left, these are human cells, and we're looking at human GAPDH. And you'll notice in the inset these 2 large, very bright spots. These are actually sites of active transcription, and you see the buildup of nascent RNAs being actively transcribed. And again, it doesn't matter what species you're using. We're looking at, in this case, now mouse GAPDH on the right, the mRNA for that. So we can detect it. We can detect RNA. And now what's also a very nice feature of Stellaris FISH is one can actually localize on the subcellular level. These are RNAs of interest. Now in this case, we're looking at the transferrin receptor messenger RNA. And unlike the GAPDH you saw earlier, it's actually located distinctly on one side of the nucleus. This is probably because it's the transferrin receptor is a membrane protein, so the RNAs are probably tethered to the ribosomes, which are being tethered to the ribosome on the rough ER, which are being actively translated into the endoplasmic reticulum. So we can detect, we can localize, and more importantly, we can quantify. In this example, we're looking at the FKBP5 messenger RNA. The target's not important, but it's just to show you that in the presence of the steroid dexamethasone, we can see a strong upregulation of the messenger RNAs of FKBP5. Now, if you look on the right, the graph on it, you'll notice you'll see about a 17-fold increase with the addition of dexamethasone, but you'll notice the the relatively broad range of, for example, 11 plus or minus 6, 183 plus or minus 60. Now, this is in fact due to, in fact, the stochastic gene expression at the single cell level. So this is something one misses with by qPCR or by northern blotting, where we're actually counting, single cells and the RNAs in each single cell. Hence the relatively broad range for each bar. What's also nice with the Stellaris FISH is we can actually multiplex. So we're able to look at multiple RNAs at the same time. In this case, we're looking at the 3 different families of the NCOA family, COA 1, 2, and 3. They are on the RNA level, they're very different, so we can look at these at the same time. And I blew up the area on the right, the inset there, and circled areas in green and yellow and red to show you which ones correspond to which messenger RNA. With Stellaris FISH, it is also compatible with fluorescent reporters. In this case, we had overexpressed translinearly transfected EGFP in 293 cells. Obviously, you can see it from the green on the right, and in this case, we were actually looking at the EGFP messenger RNA. Now, for most situations, you would not be looking at the messenger RNA of EGFP. You'd probably be looking at an RNA of another target. For example, if you were to target your one protein with EGFP, and then look at RNA of a protein that may be interacting or maybe upregulated or downregulated by this tagged protein. So, just to show you that the Stellaris FISH is very much compatible with fluorescent reporters. We can also simultaneously look at the proteins as well as the RNAs at the same time. In this example, we are looking at the HER2 protein in the SKBR3 breast cancer cell line. This cell line is well known for having multiple copies of the HER2 gene, and as you can observe about 1,000 copies of mRNA copies of HER2, and you'll see the signature HER2 protein as the outlying the cell. Obviously, it's a membrane protein. Again, just remind you that the HER2 mRNA can easily be substituted by your long non-coding RNA of interest. So you could look at your long non-coding RNA and as well as look at your its protein, a protein at the same time. Now, Stellaris FISH can also be used in tissue, in both frozen and formalin-fixed paraffin embedded. And in this example, we're again looking at the HER2 messenger RNA. These are clinical samples that were classified by the pathologist to be HER2 negative on the left and HER2 positive on the right. And you'll see quite easily, the dramatic upregulation of the HER2 mRNA on the tissue that was deemed HER2 positive. So, again, we can see not only can we use Stellaris FISH in adherent cells in tissue culture, we can also use it for tissue, which is very, very nice. Now, the data we are generating, is it comparable to what's the data from the published literature? Now, what's convenient is that, again, if we're going back to the HER2 gene, the HER2 protein, in fact, in this figure was looked at in several breast cancer cell lines, but I want you to focus in on SKBR3, MDA MB231s, and the MCF7 cells. And in this work, they were able to show that a HER2 protein is, again, it's expressed most in the SKBR3 cell line. Again, and then it drops down to the MCF7 cells, and then even less in the middle in the MDA MB231 cells. So this was very good test for us to see how well does the Stellaris FISH compare to standard Western blotting? Is it consistent? So what we did was we actually took those cell lines, all three, SKBR3, MCF7, and MDA-MB231s, and when we performed the Stellaris FISH, we were able to do the counts. We were able to see this nice consistency with the published work. We see about 1,100 molecules of mRNA in SKBR3, about on average 67 molecules of mRNA in MCF7 cells, and about 20 molecules of HER2 and MDA-MB231 cells. But unlike the Western blotting, which just gives you a relative amounts, we can actually give you hard values, or the raw values on a cell-by-cell basis. So it's very, very quantitative. So obviously the next question is Stellaris FISH and long non-coding RNA detection. So this was obviously a natural transition for us to go from messenger RNA to look at long non-coding RNAs. It was a no-brainer basically. And to show you now, to give you more examples of Stellaris FISH and looking at long non-coding RNAs, we're looking at now HOTAIR, which I talked about earlier, as well as the XIST RNA on the right. And for HOTAIR, you can see that it is mostly nuclear. You can actually count this with your eye. It's very low abundant, unlike NEAT1 and MALAT1. And on the right, this is XIST. Now I should tell you first that SKBR3 cells have 3 X chromosomes. So what you're looking at is actually the XIST RNA coding the 2 inactive X chromosomes. But again, we can see a very consistent results for both HOTAIR and XIST. Now, in this example, we're now looking at the long non-coding RNAs TUG1 and the P10 pseudogene 1. You'll notice that it's much more cytoplasmic than the previous examples that we saw earlier. You see TUG1 is both intranuclear as well as cytoplasmic. And the same can be said for, actually even maybe more cytoplasmic for the P10P1 RNA. Now, I showed you this one from the beginning, but just to remind you what this looks like for the next slide, we see NEAT1 here and MALAT1, very robust expression in these lung cancer cell line. Now, I mentioned earlier that we can use Stellaris FISH for looking at tissue. And in fact, we were able to acquire mouse tissue that was either wild type for MALAT1 or knocked down for MALAT1. And again, you'll see on the left, you see this nice discrete localization of MALAT1 in the nucleus. And as we had expected, in these mice that were knocked down, knocked out, just knocked down, we see a strong reduction in the MALAT1 RNA in the nuclei. So just in summary, I just want to point out that we can easily visualize and quantify RNAs in situ with Stellaris FISH probes. Now, multiplexing, it's just merely dependent on the filters that you have. And Biosearch has a catalog of dyes that will fit your needs. Fluorescent dyes. Now, Stellaris FISH is also compatible with Immunofluorescence, as I mentioned earlier, and fluorescent protein reporters. And this tool, Stellaris FISH, also will work in frozen and paraffin-embedded tissue. And more importantly, Stellaris FISH is the only tool that allows one to look at long non-coding RNAs. There is no other tool out there right now. I just want to give some of the important links to, if you wanted more information about Stellaris RNA FISH, we have this link up above, and the second one is to the probe designer. When you just have to create a login and password, and you're good to go. You can start creating Stellaris FISH probes immediately. And here's also my contact information if you want to ask me more questions. I welcome any questions you have or comments, and I will get back to you almost immediately. And lastly, I just want to acknowledge everyone at Biosearch Technologies, from R&D to the technical support department, and as well as our marketing department, and our academic collaborators, Arjun Raj from UPenn and Sanjay Tyagi from UMDNJ. And with that, just want to I'm willing to take, or I welcome any questions you may have. Thank you very much.
Thank you very much, Arturo. That was an excellent presentation, some stunning images in there. So we already have quite a number of questions in from the audience. If anyone has another question, just post it in the box and we'll try and get to it. So let's go to these then. So the first question, Arturo, is one that's been asked by two people, Kimberly Crawford and Jin Song Kui. They're asking how many of the probes must bind to generate an individual dot?
Right, that's actually a very good question. We found that obviously it'll vary on the target, but in general, if you can generate at least, I would say, 25 Stellaris FISH probes, then your success is very high. If you can generate at least 30, it is very high. So 30 on, it's a very high chance that you'll be able to see your RNAs of interest.
Okay. So we'll just swiftly through these. Because I think we're packing as many of these as we can. So Jin Wu Li is asking, how can we prove that these probes bind to RNA and not DNA?
Oh, that's a very good question. I didn't mention for the protocol that the protocol does not have a denaturing step. So the double-stranded DNA will still stay double-stranded and will not be accessible to these Stellaris FISH probes, these 20mers. So extensive experiments were done by Arjun Raj and Sanjay Tyagi to show that these probes will not bind DNA with this protocol.
Cool. Okay. A question from Wayne Speckman. What are the dyes on the probes? Do you have deoxygenin label probes, for example?
Well, as far as we're currently offering the Stellaris FISH probe, since again the power is in the single molecule detection. It's mostly available available right now in fluorescent dyes, so they're not desoxygenin labeled.
Right. So they're just different fluorophores that you can choose from those when you order the probes, yeah.
Correct. That is, yeah. We offer again. We offer dyes that are very much compatible with the filters out there.
Yeah. Okay. Wayne's also asking, do you offer software for spot counting?
Yeah, we get this question a lot. As of right now, we have an in-house quantification software that we're trying to get out to the public as soon as possible. So the only thing I could recommend as of right now is you should use your proprietary scope software or something like available freely downloadable like ImageJ. That's the only things I can recommend right now. But again, we are trying to get this quantification software out to the public as soon as possible.
That sounds like that would be really useful. So one from Mikhail. What negative controls do you use? Can you order the sense probe for a negative control, for example, or is that what you would recommend?
Right. So we don't recommend that. I mean, we get this question again a lot. Can we just order the antisense to the probes? But we found that genes can be, they're quite often they're transcribed in the antisense direction. So, it's not gonna. It could give you discrete spots, which would be real. So our best example of a negative control would be using a cell line that for sure does not express your gene of interest. That would be the perfect negative control. You could, I suppose, also maybe a probe set to GFP or something like that, where it's just not native to the cell of interest.
Yeah. That's a good approach. One from Joe Battistelli. Do you think that this technique could be used to work on to hybridize mRNA in bacteria, for example, from environmental samples?
Oh, yeah. We actually on our website and through work done by Arjun Raj, he was able to show, and other people, I should say, other customers, they've been able to share pictures that this will work very much in bacteria. It's the protocol it's a little different, but it's very much comparable to what's been used for adherent cells and adherent cells.
So do you offer different protocols for different cells and tissues and things, or?
Yes, we do.
Yeah.
We have one for inherent cells, we had one for tissue, and we have one that encompasses one for, I believe, yeast and Drosophila. So it's on the same page, I should say. It's not the same protocol, but yes, we have compiled different protocols for your needs.
Cool. Okay. And let's see. So Wayne Speckman again is asking, can you make comparisons between probe intensities, even though the number of probes can be different, plus it isn't unknown how many actually bind?
Right. Again, that's a good question. So obviously the RNAs are not going to be all equally accessible to the same number of probes for one reason or another. There is I guess that's one of the powers of this. There's a Stellaris FISH, this built-in, this redundancy. Basically, you have 30 to 48 probes to play with. You're hoping that most of them will bind to your RNA of interest, and so as far as comparing intensities, we don't usually do that. It's more like it's more for quantification purposes. We just compare, can we count the number of spots?
Right.
And well, I should also add that,
Yeah.
the brightest spots should be in the nucleus because those are the sites of active transcription. But for the most part, we're just counting single spots that are clearly well above background.
Yeah. Okay. And what is the minimal length for a non-coding RNA to be detected?
Right. Again, it goes back to can we put at least 25 of these 20-mers onto your non-coding RNA. So there are examples though, unless they're in clusters. Now, so we are able to look at this other species of RNAs that are present in clusters and nuclei. Now we can see them with 5 of these Stellaris FISH probes because there is this amplification of all of these RNAs in one area. It's no longer quantitative, but we can at least detect them, and very easily, in fact. So it's if you get lucky and these are present in clusters in the nuclei, then yes, we can get away with even with FISH probes that you would not normally be able to use.
Yeah. Okay. So on the probe designer, Vikas Palhan's asking, does your probe designer include a blast search for specificity?
That's a good question. It's what it does is that it does, through previous work from our academic collaborators, they've been able to remove problematic sequences that would make your Stellaris FISH probes non-specific. As far as blasting, I think that would be up to the individual to do, to make sure they would want that it specifically detects their RNA. But right now, it just removes problematic sequences, not necessarily blasting.
Sure. Okay. Chen Fan Yang is asking, can your fish slides be counterstained with IHC or IF? I think you showed a couple of slides with that on there.
Yeah. It's and if that person wants to contact me on a protocol and how to do that, I could oblige.
Excellent. Okay. So just 2 more questions. Well, one is I can answer. Is quite a few people ask, are the slides available for download? So, well, the video will be definitely available for download on the Biosearch website, I believe, and on our website at bitesizebio.com. I'm not sure whether you want to make the PowerPoint available on your website, Arturo. But
we'll discuss it amongst our team.
Sure. And the final question, it's from Michael. What is an approximate cost per set, for example, for 35 probes?
So one set, whether it's a set of 12 or a set of 48, it will go for a $575, which is good though. For enough for about 200, at least 200 hybridizations.
All right. Okay, that's quite a lot of experiments for your money there.
Oh yeah, I mean you get your money's worth.
Certainly. Okay, thank you very much. We'll wrap it up there, Arturo. Thanks for a really excellent presentation and for getting through all of those questions.
Oh, thank you, Nick. Thanks, everyone.
You're welcome. And so thanks also to our sponsors, Biosearch Technologies. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the seminar and you'd like to view the video recording of the session, please visit the seminars page on bitesizebio.com, and it should be available there within the next 24 hours. And there you can also see the other webinars that we've lined up for you over the coming months. So until next time, good luck in your research and goodbye from all of us at Bitesize Bio.